Hi guys, it's me, Lauren. Welcome back to my channel. I had a lot of requests from you guys to do something a little bit more sciencey, so that's what we're going to do today, and we're going to talk about the spinal cord. So when I was doing research, there was one study that really stood out. Vahdat and colleagues actually used neuroimaging techniques to look at the spinal cord in the brain during learning to show that the spinal cord was actually involved. So I'm not sure if you guys know this, but for those of you that don't, the spinal cord consists of different areas, and today we're going to really look at the cervical spinal cord. So if you hear me say things like C6 to C8, that's me referring to that part of the spinal cord that innervates the limbs, the upper limbs, okay? So before we talk to our very special guests, first I'm going to tell you why Vahdat's article is so freaking cool, okay? So we've known for a while that the brain has been involved in learning, but we always thought the spinal cord was passive, but Vahdat and al. Actually show that the spinal cord demonstrates plasticity involved in learning. There have been animal studies that have showed plasticity in the spinal cord after basic learning through lesion and operant condition studies, but Vahdat fills this gap by looking at humans and using neuroimaging. Pretty cool. So studies on humans have looked at reflexes and how they decrease in areas that are used by highly experienced individuals. So this tells us that the spinal cord reacts less intensely once it's learned. And so Vahdat actually looked at earlier learning to see this in the spinal cord. Also, there's electrophysiology studies that show changes in the spinal cord synapses in response to learned movement. But electrophysiology studies can't differentiate from descending cerebral inputs, so the study we're looking at today measures both the brain and the spinal cord in order to measure their independence. So basically, in the past, neuroscientists have only been studying the brain in learning and assuming that the spinal cord remains passive. But this one study by Vahdat and his scientist buddies found some nifty novel results using a well-established motor learning sequence. So I'm headed to Queen's University today to talk to the scientists behind the findings. Let's go.
Here we are at the new medical building. Let's go in.
Hi guys, I'm here with Micah, who worked on the Vahdat paper.
Hey Lauren, thank you so much for joining us at Queen's today.
I'm so excited. So what was your research question?
With our research, we wanted to investigate whether human spinal cord activity at the cervical level shows intrinsic learning related changes during motor sequence learning. Our approach is novel because we used a regular fMRI scanner to measure both brain and spinal cord activity simultaneously, which has, to our level, to our knowledge, never been done before.
Interesting. So how did you collect data?
First, we recruited 25 young, healthy right-handed participants just like yourself. Later in our lab, we asked them to perform a motor sequence learning task with their non-dominant left hand. Generally, a motor sequence learning task refers to the process by which a sequence of movements come to be performed faster and more accurately than before. You could try the task yourself if you want.
Ooh, sure, I'd love to.
Our control sequence is simple. Just repeat the sequence 4, 3, 2, 1, with 4 referring to your pinky and 1 to your index finger. The second one is more complex. Try repeating 4, 1, 3, 2, 4 as quickly as you can. For our study, we used a block design with 24 blocks of 60 movements each. Each block was separated by 15 seconds rest period, allowing neural activity to return to baseline. We recorded both structural volumes and functional data. The functional acquisitions included 35 to 37 transfers and slices per volume. Moreover, we recorded the functional data from each subject's cervical cord from 15 actual slices spanning from the cervical spinal segment C1 to C8. In addition to our main sample, a separate control group of healthy young subjects completed the same task in an MRI, recording electromyography signals for both extrinsic and intrinsic hand muscle.
Why was there an additional control group?
We included the second group in order to make sure that the changes in performance speed were due to spinal cord plasticity rather than being a right byproduct of variations in applied muscle force during motor learning. Confirming that, we found no significant effect from either the simple sequence or the complex sequence in any of the tested muscles. And then after the initial experiment, we applied motion correction and spatial smoothing to make the data more comparable between subjects. To avoid noise within the data, we also applied a high-pass filter and removed non-brain tissue areas from the scans.
So what did you find?
For that, I'll refer you to my colleague. She's an expert on the results.
Hey Lauren, I'll explain what we found. The performance speeds averaged across all subjects was significantly increased in both complex sequence and simple sequence conditions over the course of learning, but an analysis of variance showed different learning curves between the two conditions, with participants reaching asymptotic performance earlier for the simple sequence than the complex sequence condition. This suggests that distinct learning mechanisms were involved during the simple sequence and complex sequence training conditions. By taking activation maps of sagittal slices, we were able to show that there is a main effective practice during the two conditions. There was greater bold activity when learning a complex motor sequence compared to a simple one, both in amplitude and spatial extent within the C6 to C8 levels of the spinal cord, ipsilateral to the left-hand finger movements. This location makes sense because it is where we find the motor neurons innervating the finger muscles involved in the motor task. Furthermore, the complex sequence condition generated significantly greater modulated activity during motor learning with a significant difference in the mean amplitude of the bold signal change and in the spatial extent of the activation within the C6 to C8 areas, indicating that learning-related behavior was improving. Yet, this approach cannot inform as to whether these changes are a simple reflection of plasticity occurring in the supraspinal sensory motor structures, meaning the brain, or whether they actually represent plasticity in the spinal cord. To explore this, we conducted a general linear model analysis to partial out the possible supraspinal contribution onto the cervical cord bold activity. Interestingly, the results revealed significant changes in cervical activities during learning in the C7 to C8 segments that were independent to the signals from the brain structures known to project to the spinal cord or that showed learning-related activity changes. This suggests, during motor learning, intrinsic local plastic changes can occur in the spinal cord, independent of the brain. Finally, we evaluated the linear correlations between bold signals of the sensory motor cortical areas and the spinal cord C6 to C8 segments. We found that the spinal cord becomes less synchronized with supraspinal sensory motor areas over the course of motor learning. A brain-to-spinal cord functional connectivity analysis demonstrated that the initial linear relationship between the spinal cord and the primary sensory motor cortex gradually becomes less correlated over the course of the complex sequence learning, while the spinal cord also becomes more negatively synchronized with the cerebellum as their inhibitory connection gains prominence. Altogether, these data suggest that the spinal cord contributes distinctively from the brain to the motor skill learning process and is much more plastic than it was assumed before. However, there exist some limitations that we were unable to address in this study and await further investigation. First, we are unsure of the exact neurophysiological mechanism underlying spinal cord neuroplasticity. Second, while it was proven that the spinal cord activation is linearly independent from supraspinal structures, this does not exclude the possibility of a non-linear relationship between activities in these two regions. Third, we are not sure what mechanisms might explain the observed pattern of connectivity between the brain and spinal cord, as they cannot be directly tested with fMRI. Future researchers will need to parse out the roles of these two structures in various stages of motor learning. Fourth, because we were interested in acquiring data covering both the brain and spinal cord, the spatial resolution in our study was limited, and we could not make conclusions on the precise location of focal activity within the spinal cord, especially in determining whether C7 and C8 act as one functional unit or two separate ones. Finally, because we acquired a limited number of volumes during the rest periods between blocks of tasks, we were unable to determine what functional changes in the cervical level of the spinal cord persist after learning the sequences.
Wow, thank you so much.
If you enjoyed this video, be sure to check out their channel, Scientists for You. I'm going to post the link in the description box below. Bye guys!
Everything we talked about today was so mind-blowing. All the research is very applicable and very novel. It takes the field to a whole new level. First of all, it showed plasticity in the spinal cord, unlike any studies, so it doesn't really fit into the majority of the research. If you think about the bigger picture, imagine if the spinal cord plasticity allows us to relearn motor function. This could improve clinical implications for rehabilitation of patients with spinal cord injuries. Because it was the first time fMRI was used to study spinal cord activity and brain activity simultaneously and using regular equipment, it opens the doors for new research. And that's all for this week. Remember to like, comment, and subscribe, and tune in next week for my video on how to clean your makeup brushes.